Metabolism. Metabolism is a sum of energy being expended and energy being stored. So energy coming in, energy going out. Most of you already knew this from your health class where you look at metabolism and calorie intake and calorie burning. And calories are a unit of measurement for metabolism in molecular biology as well. We will talk more about that. So metabolism, really when you're talking molecular biology, you're looking at a pathway. One of my favorite pathways is the cholesterol pathway because cholesterol is the beginning point of steroid synthesis. So from cholesterol, your body has a pathway to create testosterone or estrogen or cortisol-like or human growth hormone-like different steroids. So, no, in fact, pregnant women really need cholesterol because cholesterol, these guys will tell you, is part of every cell membrane, right? So, every cell membrane has cholesterol in it, and you're growing a baby who's growing lots of new cells, they need cholesterol. So,
why is it a killer?
It's a killer when it's free-floating in your bloodstream. And that's a regulation of enzymes, actually. And so, especially as you get older and you're not creating as many new cells, one reason you're just going to have a natural increase in floating around in your bloodstream because it clings to the sides of those capillaries and arteries, and that's what's the problem. Good question. Absolutely. So your body's going to regulate this all along the way. Every one of these points is a regulation point. This doesn't mean it's going to allow it. Enzymes don't allow things. Enzymes either encourage or catalyze things, or they inhibit them and get in the way of them. That's what regulation is. Does that make sense to you? Regulation is you're either making it more likely something's going to happen, or you're going to decrease how likely it is to happen. We're going to talk about examples of this as we go through here. Yeah, lots of examples. So let's just talk some basics of metabolism again, or energy. Catabolic pathways are your pathways that are breaking down complex molecules into simpler molecules, probably through what process? How do you usually break down proteins? Hydrolysis, yeah, a lot of this is happening with hydrolysis. I always think of a catapult, releasing energy. Catabolic reactions release energy. Versus anabolic, I always think of either Anna storing apples, or some of you might think of anabolic steroids, which build big muscles. So you're building up proteins from amino acids. You're building. Anabolic is building.
Catabolic is what? Release energy, and the catapult destroys them, right?
Not always. Catabolic pathways release energy by breaking down complex molecules into simpler molecules. Anabolic consume energy to build more complex molecules from simpler ones. What's another example of a anabolic pathway in living organisms? Photosynthesis, classic. You're taking energy coming in to make a more complex molecule from simpler ones being carbon dioxide, complex ones being sugars. Bioenergetics is the study of how organisms manage their energy resources. So we're going to talk about thermodynamics in a minute. That's just the study of energy, whether it's biology related or not. Bioenergetics is looking at living organisms. And some fascinating little guys like in the Sulphur Lake in Yellowstone, or down in the thermal vents in the bottom of the ocean where there's no sunlight. How are they getting energy? That's a question for a biologist who studies that. Are they using the thermal gradient? Are they using sulfur gradient, pH gradient? When I say gradient, you know what I mean, right? Because you just did membranes. You know, there's a diffusion across the gradient. Gradient is more concentrated area, less concentrated area. So if you have a thermal vent, you've got differences in temperature and pH there. So there's a lot of different forms of energy. You have known of most of them for a long time now. Kinetic energy, when we talk about it in AP biology, we're usually looking at the motion of the molecules. And so quite often when we measure the average kinetic energy of the air in this room is measured using what? A thermometer. So this is usually going to be measured with temperature. Now, you can measure kinetic energy in your physics class by rolling a ball downhill. Yes, that is also motion. This, in AP Bio, we're generally looking at temperature and how fast molecules are moving in your body. Heat, thermal energy is kinetic energy associated with random movement of atoms or molecules. I don't really like this definition. I don't think it's good enough. How is that different than kinetic? So clearly the most popular person in the room is not me right now. It's okay. I can deal with that. Heat deals not only with kinetic energy, but with how many molecules do you have doing that kinetic energy. How do we measure how many molecules we have? Mass, right? So if I have a little swimming pool I want to warm up, if my kinetic energy, my temperature, I have a boiling cup of tea. Is there a lot of heat in a boiling cup of tea? Well, yeah, but what if I want to warm up this swimming pool? I have a boiling cup of tea, or maybe I have a pretty warm, like 50 degree, which is warmer than body temperature, but it wouldn't burn you, but it's a pretty nice hot bucket of water. So I have a warm bucket of water or a boiling cup of tea? What's going to have more heat? If it's a bucket and it's a pretty massive bucket, that's going to have more heat because heat is mass and temperature. So when we look at like the heat it takes to melt an ice cube, we're going to look at how massive is that ice cube, right? Because the more massive the ice cube it is, the more heat it's going to take to melt it, right? Potential energy. Energy matter possessed because of its location or its structure. Usually structure is what we're looking at in biology. Sugar has a structure. Carbon dioxide has a structure. One of those is a more stable structure. There's not a lot of potential energy. If you're more stable, you don't have very much potential energy, right? If I stand up and stand on one leg, I have a lot of potential energy. If I jump up, that's going to immediately, I'm going to fall back down to a stable situation. Chemical energy, potential energy, this is a type of potential energy that we're always going to be talking about. Pretty straightforward. Lots of potential energy, not as much potential energy. Okay. So if we're going to look at energy overall, then you're going to talk about the first and second law of thermodynamics. And I really struggled with the second law of thermodynamics philosophically for a long time, but I finally feel like I'm okay with it. First law of thermodynamics I'm okay with, even though I know that it's a little bit fuzzy when you get into fusion reactions where mass is converted to energy and energy, it's like, wait a minute. But thermodynamics, basically, in a closed system, there's no change in thermodynamics. It's closed. Energy's not going in. Matter's not going in. Is the Earth a closed system? Is our planet closed? No. What's coming into our planet? So we've got sunlight coming in. Do we have anything else coming in? A tiny little fraction of asteroids and cosmic dust. But really, matter is not, our Earth for the most part is a closed system as far as matter goes. We're stuck with everything here. You think you're throwing something away, and it's going in the garbage and it's going away, but you know it's really not going anywhere, right? It's still on the planet. So organisms like you are an open system with, you're not like the planet. Stuff comes in, stuff goes out, and it's totally changed. And you leave it behind, and you don't carry it around with you everywhere you go. Yeah?
Back to the picture with like the diver, like the
Yeah, we can go back to the picture. Yep.
Isn't the person in the water, wouldn't he have more when he heading in water?
He might have a higher kinetic energy at this point, but not a greater potential.
Oh, okay.
Good question. Okay, first law. Energy is not transferred, or it is transferred or transformed, but it's never created or destroyed. That's the law of conservation of energy, which most of you are familiar with. Now you're going to become more familiar with the second law of thermodynamics, is that every energy transformation is increasing the entropy of the universe. What is entropy? Disorder or messiness. Or randomness. Randomness is a better word because then I can think about how quickly molecules are moving. So I could take ice, liquid water, and water vapor. And which one has the greatest entropy? Or randomness? The water vapor, right? Here you see chemical energy being transformed into heat and energy to run reactions. You also see this is probably a nice, neat molecule, and now it's broken up into water vapor and carbon dioxide, which is increased messiness. And it's like when I look at that example, I go, okay, that follows the second law of thermodynamics. I'm going to come back to this. But what about when you're dealing with a plant? They're taking light and making more complex molecules. Isn't that disobeying the second law of thermodynamics? Because they're definitely decreasing entropy right there. Yes, they are decreasing entropy within themselves, within the situation. But the second law of thermodynamics is that for these reactions to happen, you're increasing the entropy of the universe. And that still is happening because that sunlight is being converted into heat, even for these plants. Do we have to go back one more? We're going to spend a good 5 minutes or so talking about whether or not a process is spontaneous. And the reason it's important is because your cells work, because they couple a spontaneous process with something that's really hard to do. And you could think of it as, if I want to get this rock pulled halfway up the hill, I'm going to tie a smaller or similar sized rock that's already at the top of the hill. I'm going to throw it down, and it's going to pull up the other rock with a pulley system. And that way you've coupled a reaction. And I'll show you a great example of that in here. So cells overall are creating more ordered structures from less ordered materials, except for you consumers and your sugar, where you create carbon dioxide and water vapor from your sugar. But that's why they say down here in the second bullet, you're also replacing ordered forms with less ordered forms. And they give that example of sugar and water. Where does the major source of energy come to our planet from? And the sun. And what's an ecosystem? An ecosystem is, in a given area, all the living things and the non-living things and how they're interacting. Okay, let's talk about spontaneity. There's this guy named Gibbs. And if you have your equation sheet, there is an equation to calculate Gibbs free energy. Gibbs did some measurements and figured out how to predict whether a process was going to be spontaneous or not. So, free energy is energy that's available to do work. Okay? And in order to be spontaneous, you have to be rolling down the hill. You have to have a negative free energy change from reactants to products. Does that make sense? It's not going to be spontaneous for me to push a rock up a hill. I have to put energy into that system. It's not going to be spontaneous. For a process to be spontaneous, it has to have a negative free energy change. So I think I can go one more before we talk about this. Yeah, let's sit here a minute. So what is delta G? Free energy. What is delta H? Well, they say total energy. They say change in enthalpy. Enthalpy, I remember this as heat. And it's pretty safe to think of delta H as heat. So what is the change in the heat of the system, the overall heat? And then what's delta S? Entropy, or messiness. S for mess. Not as good as randomness, but it might help you remember S is for mess. So let's talk about a process that you know pretty well. Let's talk about melting ice. Yeah. So, did I take this from somebody? No. From me? What did I say? Delta G has to be in order to be spontaneous? It has to be negative? Okay, you guys are from Michigan, so you know about ice. Is ice going to spontaneously melt in this classroom? Yeah. So you're saying that the delta G is going to be negative for this. What's the change in heat going to be? Is the ice going to absorb heat? Yeah, so it's going to be positive delta H. Wow. So in order for delta G to be negative, this right here is pretty important. As it goes from solid to liquid, is it increasing in randomness? Solid to liquid. Yes, it is increasing in randomness. So our delta S is going to be positive delta S. What's our temperature at? Well, temperature has to be in Kelvin. So if in this classroom right now it's about 20 degrees Celsius. How do I convert it to Kelvin? Good, add 273, so I'm at 293. Now you have no idea what this number is. My honors chem class just did this experiment. I thought about having you guys do this experiment, but you're running kind of short on time, and you've already got enough labs going on. So I'll just tell you that to melt ice is 335 joules per gram. Now you're used to thinking of heat as calories, right? All you have to do is multiply by 4.184. It's not even an equation on here. If they're going to talk in calories, they're just going to give you a slightly different number here for the calories it takes to melt a gram of ice. They might even say it in kilocalories per mole, because what's a mole? It's important next week, because it'll be October 23rd. Oh yeah, it'll be mole day. 6 times 10 to the 23rd, or a pile. It's a very large pile of atoms. And that's what we're dealing with in our bodies, right? Our piles of atoms. So you'll sometimes see it as kilocalories per mole. This is a pretty big number. Delta S for ice melting, you guys told me it was going to be positive. I don't know what this number is, but it's going to be something. And you know that overall this delta G is going to be negative. So what changes when you try to melt the ice outside in January? This delta S is still going to be that same number. What's going to change? Temperature. It's going to be like, I don't know, negative 10 Celsius outside, below freezing. If freezing is 0, we can put it 0 if you want. If I put it 0, this is 273. This is still 335 joules per gram for the ice. And evidently, at that point, this becomes positive. How do I know delta G is positive for ice melting outside in January? Because I know that it's not going to happen spontaneous. Delta G is a measure of spontaneous. Delta G is that free energy available to do this. It's just going to roll down the hill if delta G is negative. So let me show you a picture of this. Question, Mary. Sure. Oh, my bad. Thank you very much for correcting us on that, or me on that. Should be negative there. You're always going to subtract T times delta S. Hey, what happens if delta G is 0? Nothing. It's in equilibrium. And you've seen that happen out there in the lake, where the ice is liquid at the same time it's a solid, and it takes a long time for it to really go all one way or all the other, right? You want me to restate this stuff a little bit better here? Again, free energy is your heat or total energy minus the unusable energy, because you can't really use I don't know why they say this way. I really don't. I just memorized that. Negative delta G, spontaneous, because it's like rolling down a hill. And in biology, a spontaneous process, we can couple that with a non-spontaneous process and make something happen. Question? Delta S, I was multiplying it. It's a question of, is messiness, is randomness increasing? If it is, it's a positive delta S. Let's look at a couple situations. Where'd it go? I wanted to look at a bigger version of those, but I guess I'll just look at these. We're going to see a bigger version of these soon. Can you look at these pictures and tell me, well, especially look at the last two, diffusion and chemical reaction. What's happening with the messiness or randomness of this reaction? It's increased. It's gotten more random. So this is going to have a positive delta S. It's increased in randomness. That makes sense now? They're farther apart and they're moving more quickly, and that's more random. And then in this one, you have probably some pentose sugar. And at the end of this reaction, you have carbon dioxide and water vapor. So you've got all these little molecules. So you can either increase the speed at which things are moving, will make them more random, or you can increase how many pieces there are. That will make it more random. So if I put salt in water versus putting sugar in water, if we think back to our water potential problems earlier this week, what was that ionization constant for sucrose? It was 1, because it doesn't split up into ions. And the sodium chloride, the ionization constant was 2, because it splits into ions. So which one has a greater entropy change, or messiness, or randomness change? The salt, because it's splitting into 2 pieces. So you got more pieces. Temperature is the same for both those situations. Pressure was the same, but you had increased randomness. And that changed a lot of other variables. It made it more of increasing the water potential even more, the movement of water. Okay. Did I miss something back here? Again, free energy is instability. If you're in equilibrium, that's as stable as your system can get. A process is spontaneous and can perform work only when it's moving toward equilibrium. If it's already at equilibrium, it's done. So we're going to look at exergonic versus endergonic. How is this different than exothermic and endothermic? Not much. For those of you that remember that from chemistry. The only difference is we're going to, instead of an exothermic and endothermic, what did we put on the y-axis? We put heat right here. We had delta H. We could say heat over here. And if a system is absorbing heat, we called it endothermic, because thermic is heat. And if it was, oops, that's not a very good C. And if it was losing heat, that'd be a negative delta H, and that would be exothermic. Positive delta H. So when we refer to exergonic or endergonic, the directions are the same. It's just that your axis, instead of having delta H or heat, you have free energy. Put delta G on this side and look at, is it spontaneous or not? And it's actually really easy if you can imagine the graph, because if it's going to roll downhill, it's going to be spontaneous. So I'm not sure why your textbook uses column graphs for this. It's the only place I've ever seen it. And maybe the edition 8 doesn't. But if I draw a line there, this is, oh, that's not a good line. I don't like that line. I want one of these lines. Reactants start up here and they roll down here to these more stable products. That's a negative delta G. Delta G less than 0, right? So energy has been released and used probably in your cells for work. If you're going to take energy like a plant chloroplast is going to do, store it as chemical potential energy. If you put energy in, your delta G is going to be a positive. Not going to be spontaneous unless we couple it with some spontaneous reactions, which you're going to learn in Chapter 10 in more detail than you want to know. In fact, you're going to see a system like this. Did I already go past it? Yeah, we already went past the little, did we go past the little hydroelectric dams? We did? Huh. Okay. Some processes happen spontaneously. Some don't. Your cell is going to use its awesome enzymes to couple a spontaneous process with a not spontaneous process, so that it's just like I told you with the pulley and the rock rolling down the hill to pull up another rock. Let's see how this works. They're going to use their pulley system is ATP. We need to know ATP. We don't need to know 8,4, Lauren. We need to know 8,3. Yep. All right. So, adenosine triphosphate. A ribose sugar, a nitrogenous base, and 3 phosphate groups. The nitrogenous base is adenine. Does this sound familiar to you? Sugar, phosphate, nitrogenous base. Is there some other macromolecule? DNA. Kudos, whoever said that. So it's just like a nucleotide, except you have adenine here. More like RNA, though, isn't it? Because it has a ribose sugar instead of deoxyribose sugar. Is this sounding familiar? DNA is deoxyribose. RNA is ribose nucleic acid. Maybe it's good that I reminded you of that. What's happening here? This adenosine triphosphate, the bond is being broken by water, so it is called hydrolysis. Where is it breaking it? It's breaking one of these phosphate groups off. So water is hooking here and here. And when it does this, it releases energy. It releases how much energy? Pretty useful number to know. Negative 7.3. Oh, there's that unit I was telling you guys about. Negative 7.3 kilocalories per mole.
Okay, I get Mary, Tess, Michaela, Anna, Amanda, Anna.
Why don't you just take 3 at a time?
Okay, so Mary, Tess, Michaela.
So is this a spontaneous process? Yes. Why do we know that? Because delta G is negative. You're rolling down the hill. Good. The slope, like a negative slope. Rolling down the hill, negative delta G. your change in y over your change in x is a negative number. You're going down. So you have a negative slope. Yeah? You do. I said it real quick and moved on. So I think this is important for you to note. Don't get confused here. The release of energy from the chemical change to a state of a lower free energy. You actually have this release of energy when the new bonds form, not from the breaking of the bonds. Breaking of the bonds actually does take a little bit of activation energy. Just like when I release that methane and I let that methane into this room, does it immediately react with all the oxygen? No. I have to activate it with a match. And then it causes a chain reaction of this molecule releasing enough energy to get the next reaction to run, And that energy is released when the new bonds form. It's when you hit that more stable point, whether it's carbon dioxide forming or some other molecule. It's not the breaking of the bonds. Breaking the bonds requires energy. And then our room is on fire, yeah. So hydrolysis of ATP can perform work. What work do we need to do? We've got to move stuff around. We've got to do chemical synthesis or transport. What's exergonic again? Energy from exergonic reaction of ATP. Exergonic means you're releasing free energy, the negative delta G. It's exergonic. You're going downhill and releasing that energy out to the cell. Okay. This is my favorite. Here's your example with numbers, Nikita, of a coupled reaction. So what you have, glutamic acid and ammonia. Glutamic acid and glutamine, important for athletes. You guys synthesize a lot of it yourselves. I mean, humans are perfectly capable of synthesizing this. This is not a essential amino acid that you have to eat in your diet. However, some athletes actually just make sure they take extra glutamine. It's kind of controversial whether that's a really good idea or not, because it's very useful in your muscles. It's also really useful in kidneys, because this is where your ammonia is going to get secreted as urine. So that's where you're going to find a lot of this stuff, just telling you, because there it is, glutamic acid and glutamine. I thought you might want to know where it plays a role. You have to look at your protein shake. Protein shakes are quite often not that picky about which amino acids are present. They just take the liquid off the yogurt or cheese, dehydrate it, and there's your whey. So curds and whey, that Miss Moffat who said on Tough at eating her curds and whey, that's just cottage cheese. And it's just the watery substance is rich in protein. So when they make cheeses, all that milk is being digested down, and you have all this leftover liquid whey. They just dehydrate the water out of it, and they have this powder that they then put in a protein shake. It's a little processed. Well, it's processed, really. Glutamic acid. If we try to run this process without anything else, your delta G is plus 3.4 kilocalories per mole. You're pushing it up the hill, right? So it's not going to happen spontaneous. Your body knows this, finds that if it can take the ATP and make a phosphorylated intermediate, a phosphorylated glutamic acid, it's a way to couple this reaction so that now, remember that process where ATP rolled down and through hydrolysis released one of those phosphate groups and formed ADP, which is more stable? That was a negative delta G. It was negative 7.3. So if we can couple the positive 3.4 process with a negative 7, we've more than paid for this reaction. Do you see that? If I can couple this reaction of glutamic acid plus ammonia making glutamine, which costs energy, with this reaction right here where ATP is transferred into ADP, which releases or pays negative 7.3 in free energy, I still have energy left over. Woohoo! Yeah. How is it coupled? This is literally, this phosphate group hooks onto this protein. It's actually hydrolyzed by the protein. This process right here where you get rid of, when you phosphorylate it on here, there's some hydroxyl group on that protein because amino acids have carboxyl group. Carboxyl group is an HOH, so it acts like water and grabs that phosphate group. This right here, over here, this is this glutamic acid reacting with a phosphate group of the ATP and pulling a phosphate group off the ATP instead of water doing it. The water is going to come into play, actually, whoa, right here. Your water is going to actually pull this off. They show, or ammonia. Ammonia is going to pull this off. Ammonia is going to act as the water and pull the phosphate off. This is a protein. This, it has been made now because the ammonia replaced the phosphate. Yeah, this is what happens in A. This is two setups. When I run it this way, it is two separate times. If I run it this way, it's like in a beaker without ATP, I have to add energy. If I run it the same reaction with a new amino acid, this is a new amino acid. I'm doing this again with ATP. No, this is not step 1, step 2, step 3. That's why they lettered it A, B, C, because there's 3 different examples of running this reaction. If you couple it with ATP and you do this phosphorylation in the middle, it ends up giving you a net delta G of negative 3.9. Because the process of hooking on here and creating this unstable phosphorylated intermediate and then stabilizing it by adding the ammonia and ending up with ADP, you can just add these delta Gs. Delta G of turning ATP to ADP, negative 7.3. Delta G of doing it this way without any ATP is 3.4. So if I combine these two numbers, 3 positive 3.4 and negative 7.3, I get negative 3.9 overall. The difference between B and C is that they're just dropping the intermediate. They really didn't, as far as I'm concerned, they didn't need to show you C. Because what they did here, they just take this out. They just took that step out because it's an intermediate step. So when you want to look at the net reaction happening overall, you don't have to include the intermediate. 3.4 is still is this, to get glutamic acid to bond with ammonia takes that much energy. This really it looks more like it's coming from A. Step 1 and step 2. Those really are steps. So in step 1, this glutamic acid bonds with ATP instead of just trying to flat out go right to ammonia. It's kind of neither. I don't know what it is. It probably has its own number. They just didn't want to share 5 numbers with you. They thought 2 was enough. But this process right here probably has its own delta G. And my guess is it's some sort of negative delta G. It's probably pretty spontaneous. This process right here might be the spontaneous part, and this one might be positive. I don't know. They didn't give us those numbers. Good question, though. Kerr. We good with this?
Wait, can I check out Toledo?
Page 150 in our textbook, with more explanations than what you see on the slide. Full sentences, in case mine aren't cutting it, and I'm okay with that. This reaction, if you run it without ATP, takes energy. You got to push it up the hill. If you couple it with this phosphorylated intermediate and then end up with ADP being formed, ATP forming ADP is always a downhill. It's always releasing free energy. In this case, it's releasing the free energy to make this possible, to make this cost paid for. Yep, making ADP from ATP is always a downhill process. You are using some hydrogen group or hydroxyl group to pull that phosphate group off. If we go back and look at this, yes, that is why. Adding ATP, hooking ATP onto some other protein or molecule, is that what you're saying? You're asking me about? It's adding instability, because now you have an unstable situation. This phosphate, because phosphates they're pretty reactive. This is pretty, you've got all these oxygen groups here, very polar. This is more stable.
So basically that's like a molecular match.
It's a molecular match. It's a good way to look at it. Yep. Someday. And then we talked a lot about this phosphorylated intermediate that you have. This is how your cell makes things happen. It couples that ATP molecule to some intermediate, which then when it uncouples releases lots of energy to allow ion channels to pump stuff across, our favorite little kinesin to walk along that cytoskeleton like a drunken soldier, carrying stuff. We are going to spend, well, how is this ATP regenerated from ADP? Is that your question? It should be, because we're going to spend the next chapter talking about that answer, because it takes a long time to understand that. It's pretty cool. We have a really good understanding of it now. Just know that it's going to happen. It's called cellular respiration, right? This is the basics of it. We had a little, remember how we had that little energy left over? If we could have a little bit more, we could get that 7.3 we needed to make ATP again. ATP reacts with water to hydrolyze and release energy for work, and you're left with ADP. Yes, Lauren, you don't really have to know 8.4 in great detail because we assume you already know it. If you don't know lock and key method of enzymes because you don't feel like you remember it from biology, you might want to look through that chapter, that part of the chapter a little bit more. Just so you understand how enzymes work. Enzymes are a catalyst. They lower the activation energy. They make it so that less of a match is necessary to make a reaction happen. So, right here. If we've got reactants, we've got products. It is going to happen spontaneously after you activate it, and now we've lowered that activation energy. It allows reactions that would happen randomly in a flask to happen 10,000 times faster. It allows you to live. You couldn't inhale and exhale and get enough carbon dioxide and oxygen across those membranes if you didn't have enzymes lowering the activation process. So we get to go through all of this and not worry about it too much. But do know that enzymes have an optimum pH. Pepsin. Pepsin digests what based on its name? What would you guess that enzyme works on? Pepsin. peptide bonds. And peptide bonds build what macromolecules? Proteins. And if pepsin likes to work at a pH of 2, you're probably going to find it in your stomach, and it's going to hydrolyze proteins and break apart peptide bonds. How do you regulate enzymes then? This is so cool. Nope, not that part. That part. You always have oscillation between an active shape and an inactive shape. If you have an activator, it's going to stabilize that active form and allow and encourage this to happen. If you have inhibitors floating around, they're going to lock into this enzyme and change its shape to this inactive form. That's called allosteric because it's shape. Steric is shape. So you're changing the shape.
What regulates the activator?
Other enzymes? Good question. For example, let's look at this system. This is a negative feedback loop where you have threonine, an amino acid, coming in here, and then you have to do this to it, and you got to do that to it, and you got to click, 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 and then you end up with isoleucine. Now, if your cell needs to make isoleucine, that isoleucine is going to go off and be used somewhere, right? But what if it's not being used anywhere? It's a waste of energy to run this process, isn't it? So if that isoleucine is floating around, not being used, it's going to hook into this enzyme that was at the beginning. It's going to stop the whole process. And that's a negative feedback loop.